So you can see in this slide there are 4 causes. They are given 4 causes for the folding of DNA. When you see this cause, you already know the big DNA molecule, the 2-meter DNA molecule has to fit into 10 micrometer of diameter of nucleus. Okay. Now this is why the cell has to, this DNA has to fold. This is one reason. Okay. But for our purpose, for understanding purpose of the whole genetics, this is the most important point. So you need to understand folding stabilizes the DNA, that protects DNA from damage. Now DNA, there are 2 mechanisms that protects DNA from damage. One mechanism is folding. The protein, histone protein and other non-histone protein that are utilized to fold, this folding plus DNA repair mechanism. These two mechanism that protects DNA inside our body protects our DNA from injury. Now let's see why DNA is not that very stable molecule. If you read here, completely naked DNA molecule are relatively unstable in cell. And this is the reason, during DNA replication, when DNA completely unfolds, that time there is high chance that DNA will break into 2. There are double strand break in the DNA, and thanks to the mechanism of double strand break repair mechanism, our body is able to repair those damaged DNA, and we don't experience any damage. And if it is not repaired, the cell will die through apoptosis. Okay, this is the mechanism. This is the reason DNA has to be folded. DNA is folded for stability. Now, why unfolded DNA is very unstable? The question is why unfolded DNA is very stable? The answer is DNA is a macromolecule and it is anabolic molecule. Anabolic molecule. And it contains very high amount of energy. You can imagine, you can see that during replication, when you see replication process, there you can find the substrate. What substrate is required? Substrate required is DATP, DAGTP, DTTP, DCTP. These are the molecules that are added here. So very high amount of energy required to drive the whole DNA synthesis, and that DNA contains high amount of energy, and this high energy molecule tries to be stabilized anyway. So this, how it becomes stabilized? By breaking down into smaller pieces. So this DNA, any molecule that has high energy content, are thermodynamically unstable molecules. So DNA is very unstable molecule in that sense, and so we need to stabilize this DNA. And this point is very, very important for us to understand genetics. That is the reason DNA is folded, and DNA has to be folded. If DNA is not used, it has to be folded. Let's say some DNA in some cell, muscle cell, there are sets of DNA that is utilized, and there are other set of DNA that are not utilized. So those set of DNA that will be folded in compact way, we call them heterochromatin. Heterochromatin. Those DNA which are unfolded, we call them euchromatin. Only DNA which are required for the cell is unfolded. So this is the reason why DNA is folded. Now let's see then. Now when DNA is folded, it is very stable and it can fit in cell. Now the DNA can be transmitted to next generation without any damage, and then it also regulates accessibility of the DNA. So when folding, no excess. When there is less folding, the access to DNA for transcription. Okay, so the DNA which is not required for the cell, that are made inaccessible to prevent damage to those DNA. So these are the reasons why DNA are folded. And for folding, you have to remember simple concept that it requires lots and lots of proteins. The main protein is histone.